Hello, this paper presents histological classification of non-small cell lung cancer with RNA-Seq data using machine learning models. The authors of this paper are Robert B. Leshan, Mohammed Karam Moni Rabi, AKM Kamral Islam, and Marwan U. Bikdash. This research was conducted by the Department of Computational Data Science and Engineering and the Department of Electrical and Computer Engineering at North Carolina A&T State University, USA. This presentation is structured as follows. We'll begin with an introduction to NSCLC and the motivation for using RNA-Seq and ML. Then, a brief literature survey of related work. We'll outline our goals and objectives, describe the RNA-Seq dataset used, explain the proposed approach including PCA, present experimental results, compare models, and end with conclusions. This roadmap will guide us through the methodology and key findings of our study. Lung cancer is the leading cause of cancer deaths globally, with NSCLC accounting for 85% of cases. It is mainly divided into adenocarcinoma and squamous cell carcinoma, each with distinct genetic and morphological profiles. Traditional diagnosis can be challenging and subjective. Our study uses RNA-Seq data and ML to develop an objective, accurate diagnostic model for subtype classification. Prior research shows ML and DL models can accurately classify cancer types using imaging, radiomics, and genomic data. RNA-Seq provides high-resolution transcriptomic data, revealing biomarkers not detectable by microarrays. Studies by Johnson et al. And Ramroach et al. Show ensemble methods achieve high accuracy in cancer classification with RNA-Seq data, even with reduced gene sets. This slide outlines the three primary objectives of our study. First, we aim to apply principal component analysis, PCA, to reduce the dimensionality of the RNA-Seq dataset. By transforming over 22,000 gene features into a smaller set of principal components, we sought to decrease model complexity, mitigate overfitting, and improve computational efficiency, all while retaining the essential biological variants needed to distinguish between adenocarcinoma and squamous cell carcinoma. Second, we employed supervised classification algorithms to build predictive models capable of accurately differentiating between these two NSCLC subtypes. We trained and tested both classical and ensemble machine learning methods to determine which approaches are most effective for this genomic classification task. Finally, we conducted a comprehensive performance evaluation and comparison across all models, with and without PCA. This allowed us to identify the optimal classifier, or combination of classifiers, based on metrics such as accuracy, AUC, and F1 score. Together, these objectives guided our workflow toward building a reliable, interpretable, and clinically applicable machine learning pipeline for NSCLC subtyping using transcriptomic data. We used the GEO dataset GSE103584 for 127 NSCLC samples, 96 ADC, 31 SCC, with 22,126 gene features. Number samples were removed. The data was split 65/35 into training and test sets, stratified by subtype to ensure balance. Our proposed approach follows a structured machine learning pipeline designed to classify NSCLC subtypes from high-dimensional RNA-Seq data. First, we pre-processed the RNA-Seq dataset by removing non-relevant samples and imputing missing values. Next, we split the data into training, 65%, and testing, 35%, sets, ensuring stratification by subtype to maintain class balance. We then applied principal component analysis, PCA, separately to the training and test sets, reducing the 22,126 gene features to just 4 principal components that capture 95% of the variance. On the training set, we performed hyperparameter tuning using grid search and evaluated model performance with 10-fold cross-validation. We trained and compared 9 classifiers, 5 classical models, logistic regression, SVM, naive Bayes, KNN, decision tree, and 4 ensemble methods, random forest, XGBoost, bagging, AdaBoost. Finally, the optimized models were applied to the transformed test set to evaluate their real-world diagnostic performance using metrics such as accuracy, AUC, and F1 score. PCA reduced the 22,126 gene features to just 4 principal components, explaining 95% of the variance. This reduced computational cost, mitigated overfitting, and allowed us to test whether a compact feature set could still support high-accuracy classification. On the test set, decision tree and bagging achieved 100% accuracy with PCA. AdaBoost reached 95.56% without PCA. PCA improved generalization for some models, though others performed better with full features. Overall, ensemble methods showed strong discriminative power. This slide summarizes model performance on the training data, both with and without PCA. As shown in the table, models like SVM and logistic regression maintained high accuracy above 90% when using all 22,126 features. However, when we applied PCA, reducing the data to only 4 components, some models experienced a decline. For example, naive Bayes dropped significantly to 41.4% accuracy with PCA, indicating it is less effective with reduced dimensionality data in this context. Notably, ensemble methods such as AdaBoost and Random Forest remained relatively robust, retaining over 80% accuracy even after PCA. This suggests that ensemble techniques are more resilient to feature reduction and less prone to overfitting, which aligns with findings in the literature on high-dimensional genomic data. This slide highlights the performance of our trained models on the independent test set, which provides the true measure of generalization. The results show 2 key trends. First, decision tree and bagging achieved perfect 100% accuracy when using PCA-reduced features. This is a significant finding, as it demonstrates that with proper dimensionality reduction, simple and ensemble tree-based models can achieve flawless classification on unseen data. Second, ensemble methods like AdaBoost performed strongly even without PCA, reaching 95.56% accuracy, indicating robustness in high-dimensional space. We also observed that PCA improved performance for some models, like decision tree, which gained over 10% in accuracy, while others, such as logistic regression and SVM, saw a decline. These results underscore that feature reduction via PCA can enhance model generalizability for certain algorithms, particularly tree-based and ensemble methods, making them both accurate and efficient for clinical diagnostic support. A visual comparison of model performance is provided through confusion matrices and ROC curves. Confusion matrices for decision tree, bagging, and AdaBoost show how each model classified the 45 test samples, 30 adenocarcinomas and 15 squamous cell carcinomas. Both decision tree and bagging achieved perfect classification with PCA, correctly identifying every sample. AdaBoost also performed strongly with only a few misclassifications. The ROC curves compare classic versus ensemble models. The ensemble methods consistently achieved higher AUC values, with an average AUC of 0.86 compared to 0.72 for classic models. The decision tree and bagging classifiers reached an AUC of 1.0, indicating perfect separability between subtypes. These results clearly show that tree-based and ensemble methods, particularly when paired with PCA, are highly effective for RNA-Seq-based classification of NSCLC, offering both high accuracy and robustness in a reduced feature space. In conclusion, our study demonstrates that machine learning models applied to RNA-Seq data can effectively and accurately classify the 2 major subtypes of non-small cell lung cancer, adenocarcinoma and squamous cell carcinoma. The decision tree and bagging models achieved perfect 100% accuracy on the test set when using PCA-reduced features, while AdaBoost also delivered strong performance at 95.56% accuracy without PCA. These results confirm that ensemble and tree-based methods, particularly when combined with dimensionality reduction via PCA, provide a robust, efficient framework for genomic-based diagnosis. A key takeaway is that we can maintain high diagnostic accuracy with only 4 principal components, greatly reducing computational cost and model complexity compared to using all 22,000+ genes. Our study had one limitation: we excluded the not otherwise specified numbers samples. In future work, we plan to include numbers cases and extend the pipeline to deep learning models to further enhance classification performance and clinical applicability. Ultimately, this research supports the integration of RNA-Seq and machine learning as a reliable, automated tool for precise histological subtyping of NSCLC, which can assist pathologists and improve diagnostic consistency. This work builds upon significant prior research in genomics, bioinformatics, and machine learning. The references cited include foundational studies in RNA-Seq analysis, NSCLC subtyping, and ML applications in oncology. The development of histological classification of non-small cell lung cancer with RNA-Seq data using machine learning models is an ongoing process, and we welcome any suggestions or insights. If you have any questions regarding this work or are interested in collaborating on future research in this area, please feel free to contact the authors via the provided email addresses. Thank you for your attention and interest.